Hi, everyone. Thanks so much for having me. Good morning from Maryland. Today, I am going to talk to you about the vaginal microbiome, what we're doing with it at Cosmos ID, and what's going on in the field. I'm Kelly Moffat. I'm Director of Microbiome Health at Cosmos ID. I've been working here for about 9 years, and I'm very excited with where the microbiome has come so far and where it's going. Just an overview about Cosmos ID, we do end-to-end sample to sequencing to bioinformatics, or any pieces of that individually as well. We do human-based analysis, we do agricultural, basically anything where you can collect microbes in some way, even from the air. We can sequence and do metagenomic analysis of the DNA. So what is the vaginal microbiome? It is an analysis of the collection of bacteria, fungi, protists, viruses, and we can even look at antimicrobial resistance. There's even more to it than that, but this is just high-level overview. But it's basically looking at the microbes that are found in the vagina and characterizing them and trying to understand what they might be doing there. And why do we care about it? Well, we all know that vaginal health is important, I think. And we know that many women have recurrent infections, things like bacterial vaginosis, which I just will generally refer to as BV here, and candidiasis, which is a yeast infection, or it's a fungal infection. And so given this, we think it's very important to get a better understanding of what might be going on. And we can do this now by looking at the microbiome. So just some statistics, some studies, including some by the US CDC, show that up to 84% of women who have this bacterial vaginosis, which is a dysbiotic condition, don't have symptoms. And so this is something that we can see better when we do DNA sequencing and analysis of that microbiome. The healthy vaginal microbiome is lactobacillus dominant. So this is a bacterium that produces lactic acid, which helps lower the pH, which is really important because you want to have an acidic pH in the vagina, because you want to try to help prevent harmful microbes from passing further into the reproductive tract. So the risks of an unhealthy microbiome include the bacterial vaginosis, yeast infections, an increased risk of sexually transmitted infections or STIs. There's risks in pregnancy, including preterm birth and a few other risks as well. And we are also just looking at why, if you're pregnant and some of your microbiome is going to be passed on to your infant, that it's important to have it be as healthy as possible. So what can we look at when we analyze the vaginal microbiome? So if we use some methods that I'm going to get into, there's many things that we can see. So beyond just bacteria, we can look at fungi, protists, viruses, antimicrobial resistance. So this is the genes that the bacteria carry that make them resistant to antibiotics. Virulence factors, these can make microbes possibly pathogenic or more pathogenic. And then we can also do functional analysis. So this is looking at genes and pathways that are found in the microbiome. So in this figure here from a study from 2020 shows some differences in the functional genes and pathways that are found in women who have low inflammation in their genital tract versus high inflammation. So when you do certain methods of microbiome analysis, you get this information. So how do we do this? Like, what's behind the scenes? We first need to collect a vaginal sample. As you saw from the previous talk by Jill, there's a number of methods, and I'm just going to mention a couple of them a little later. And once we have the swab, we are going to extract that DNA in the lab, and then we're going to sequence it. So the way that this works is we are trying to get as much microbial DNA as possible when we break open the cells and extract that DNA, and then it gets sequenced. And this is the part that gets into the algorithms and the bioinformatics, but an oversimplified view is the way that we do this identification of which microbes are found in a sample like this. We start with a reference library. So we've sequenced a lot of different microbes already as isolates in the lab, and the whole world does this. And usually people deposit these into a big database. So we take that information to build our own library. So we know what we're looking for, because each of these microbes' genomes, all of their DNA is different. Some of them are more different than others, but that's one of the fun challenges with my job. So what we're doing is trying to take a new sample that comes in, so like a microbiome sample, and we look at this DNA in the computer, of course, and what we want to do is try to match the DNA to the microbes that it came from. And it is something that is always being refined and worked on, but it's a pretty, we've gotten pretty far with our ability to do this now. So after we've identified what's in the microbiome, we want to look at some more things. So we want to see how much of each organism is present. This is very important in the vaginal microbiome. How many different organisms are here? So this is called alpha diversity. How similar or dissimilar are particular samples when they're compared to each other if we're looking at a couple of different groups of samples? Are there any organisms present that indicate wellness? Are there any like bad players, maybe pathogens or microbes that came from another body location that have ended up in this particular microbiome? And we also look at something called the vaginal community state type. So first, how much of each organism is present? In the vaginal microbiome, we actually, for a healthy microbiome, tend to see one very dominant species of usually lactobacillus. So on the right here would be what I would consider a pretty typical and healthy microbiome for a vaginal microbiome. So it's, as you can see in that donut, it's that red area, that's Lactobacillus crispatus, which is one of the more healthy species that's found in the microbiome. And then and the abundance of that is 97%. I know it's probably small to read for you, but it's quite high, which is good. We usually like to see that. On the left is a dysbiotic microbiome where you have Gardnerella vaginalis as the top call, followed by Streptococcus, Atopobium, and so forth, E. coli. This to me would be a microbiome that does not look particularly healthy from the outset. And then we also look at alpha diversity and beta diversity. So alpha diversity, as I mentioned, is how many different microbes are present in my sample. And then for beta diversity, we're comparing a lot of samples together, and we're just seeing how dissimilar are they. So like the top two circles, they share a lot of microbes, so they're very similar, but the top ones from the bottom ones are quite different. And so what we do is plot that in a graph, and I'll show you one of those in a minute. Then we want to look at organisms that may indicate wellness. So here we have commensal, or just bacteria that are generally found in a healthy microbiome, and we list those. And then we have, so we have two different samples here, one that has a lot of diversity and one that just has a little bit of these lactobacillus species. And then we have opportunistic bacteria. So these are some where, you know, they're probably okay, but it's possible that they can cause dysbiosis if they're found in high amounts, or if they're found among other bacteria that are maybe not playing a great role. And then pathobionts, these would be bacteria that came from your gut microbiome or your stool and entered the genital tract. Probably in low numbers, they're not too concerning, but sometimes when you get to a high relative abundance for those, it's concerning. And then we have opportunistic fungi. So most vaginal microbiomes have a small amount of candida and doesn't cause problems, but as many people know, if you're a woman and you've been on antibiotics, you may have experienced a yeast infection that would generally be caused by this. And then we get into the vaginal community state types. This was all determined in 2011 by one of my former colleagues, Jacques Ravel. And his group looked at many different microbiomes over many different ethnic groups and characterized these types that they found to be the most frequent types of microbiomes. So they're basically determined by different lactobacillus species for types 1, 2, 3, and 5. And then for type 4, we have non-lactobacillus. So if you remember from earlier, we like to see lactobacillus. It tends to create a really good environment that helps keep the acidity level at a place where the microbiome is protective. But for type 4, this is going to be non-lactobacillus dominant. And we tend to see Gardnerella as one of the main genuses that's present there. So if you look here, though, it's interesting when you break it down by ethnic groups, you see different percentages of these. So it's really important that we collect this information when we're looking at microbiomes. We don't want to make assumptions based on what may be healthy for one group and not for another. Another important thing is age. So just keeping that in mind. So one thing that you might hear about is 16S sequencing, whole genome shotgun sequencing, and shallow whole genome shotgun sequencing. If you're not already familiar with it, it probably sounds like a lot, but there's similar methods, but taking a different approach. It's all DNA based, but 16S looks at only one small region in the bacterial genome. It's a very important, highly conserved bacterial gene or set of genes that codes for ribosomal RNA subunits, which are very critically important for life for bacteria. And so the premise is that you amplify only this one region. And it's great because it's affordable and it's been used in many of the studies that have led up to where we are and to our current understanding of the microbiome. You can't get the same resolution with it as you do with whole genome shotgun, where we can get strain level information. But you do get meaningful and useful information as well. So this chart just kind of gives you an idea of what we see with the different methods. At Cosmos ID, we prefer to use WGS or whole genome shotgun. We prefer to not do shallow, but to get plenty of detail as much as we can, usually up to like 20 million reads. Just to highlight 16S approaches, this is just looking at 1,000 bases or usually less than that of an entire genome. Bacteria have circular chromosomes, so that's why it looks like that. And then with whole genome, if we're lucky and we have a lot of abundance of a microbe, we can sequence the full genome in a metagenomic sample. And why do we care about strain level? There's been a number of studies that look at why do we see some women who have Lactobacillus crispatus, for example, who have a very healthy microbiome as far as it looks, may have dysbiosis. Well, certain strains may not be as good as others. And so this is where the deeper sequencing really helps us pull those apart. And also with Gardnerella, there are these different distinct phylogenetic clades, which is on the right, these groups. And so only with strain level analysis can we place these into these groups and try to learn more about what they're doing. So this reiterates the earlier point that microbiomes are distinct based on geographical location. So here we just did, this is beta diversity, that thing where we're comparing the distance based on differences of these different samples. So you see like the green ones are from Sweden, purple from China, and the blue ones are from the United States. And they really do group closely together based on those locations. And then same with alpha diversity. This is a box plot showing the distribution of the alpha diversity in the different cohorts. And in China, you have a lot more alpha diversity than in Sweden, for example. So that's something that's interesting to think about when we can't make broad assumptions. And then here, this is an analysis of vaginal samples that we've done at Cosmos ID. And we're looking at here, what are the dominant community state types? And so we're seeing one is the most frequently found, and then we go down, and interestingly, we do have a non-state type Lactobacillus acidophilus group where that is dominant, which is that fourth one down. And interestingly, that's a probiotic that can be used to help vaginal wellness. So maybe these women were using that probiotic, or maybe it just is naturally occurring in their microbiome. And then just to touch briefly on sampling, we basically, for kits that we send out to clients, we send a swab and some instructions on how to collect the sample. It's pretty straightforward and simple. After collecting the sample, you just put it into a tube that preserves the microbial DNA. So we have a couple of different recommendations here. These are really great because they keep everything stable at room temperature, so you can ship the sample through the mail and not have to worry about it being refrigerated or anything like that. So what's next? It's many things that I'm very excited about. The more data we collect, the more of these analyses we can do. But everything from pregnancy microbiome to different cancer microbiomes, STIs, HPV, endometriosis, inflammation, antibiotic use, infertility. I could probably come up with 50 things that I would be thrilled to participate in. And then personal microbiome testing has really been taking off recently, at least for Cosmos ID. People find that it's convenient. They can monitor their microbiome over time. They can start to see indications that maybe things are not as healthy as they'd like. And we're able to look at all the microbes, not just like traditional testing where you might hypothesize that a microbe is present and only test for that one. And we're also able to do really interesting research. Some disadvantages are it's not a diagnostic test. So anything we find is for research use and for using for your own information. And then also some of the methods may yield slightly different results. So it's really important to understand which type of test you have and what the expectations are. So that's all I have. Thanks so much for having me, and feel free to email me beyond this if you ever have any questions.